Polymerase chain reaction in modern disease detection. Swedish genetic anthropologist Svante Pääbo has contributed major advances to the field of evolutionary genetics. In 1992, he was awarded the Gottfried Wilhelm Leibniz Prize, which is the highest German research award. Time magazine named him one of 2007's most influential people for his ambitions to reconstruct the Neanderthal genome. In 2014, Pääbo published his book, Neanderthal Man, in which he reflects on his life and career. Utilizing PCR techniques, Pääbo sequenced the DNA of many ancient and extinct organisms, including mammoths, ground sloths, a man preserved in ice, and a Neanderthal. PCR is a way to copy a large amount of DNA from a small sample strand. It has three main steps. The first step is denaturation, where DNA is melted at high temperatures to separate the strands of the double helix. Next, in the annealing phase, primers are attached to the separated strands. In the final phase, the DNA is elongated when DNA polymerase attaches matching base pairs to the empty spots on the primed strands. This process allows scientists to analyze even very small amounts of DNA efficiently.
Let's take a look at how PCR can help people with leukemia. Acute myeloid leukemia is a cancer that starts in the soft tissue inside the bone. It can spread throughout the body to the liver, spleen, lymph nodes, and to the central nervous system. PCR can be useful because it can be a preemptive test for people who are in remission after AML. It can make sure they stay cancer-free. The testing is quickly and can be done without invasive measures. So if there is an abnormality, it can be detected by doctors quickly and addressed. PCR can also be useful in prenatal diseases. The detection of prenatal diseases have been around for a fairly long time, but the procedures are often invasive. For example, the doctors used to have to use the amniotic fluid around the baby for testing. Some prenatal diseases that can be passed from the parent to the child include an F1, sickle cell, hemophilia, Down syndrome, and trisomy 13 or trisomy 18. Now all these genetic diseases can be tested using PCR, which provides a more stress-free, non-invasive procedure.
PCR also helps with the detection of viruses that are becoming a large international concern. Viruses such as African chikungunya, Zika, and dengue have been present in the news. These viruses are all present in the same geographical locations and present similar symptoms, and they are spread through mosquitoes that contain the virus. These symptoms are difficult to quantify what disease you have. PCR can help because it allows scientists to conduct disease management and epidemiological surveys to determine which disease is present in a person. And it also allows scientists and doctors to monitor the spread of viruses through virus genotyping with PCR for relatively low cost. New techniques have also been developed regarding PCR. One of the new techniques is called quantitative PCR. Quantitative PCR follows the same steps as traditional PCR, but it adds an addition of fluorescent marked DNA and is carried out in a device called a thermal cylinder with detectors to measure fluorescence. This allows the generation rate of DNA to be measured in real time by detecting fluorescence at each PCR cycle. This data can be analyzed by computer software to calculate information such as relative gene expression in several samples. qPCR is used to quantify microorganisms that cause diseases, such as bacteria, viruses, or fungus. qPCR also allows manual labor in conducting PCR testing to be reduced because it eliminates the need for agarose gel, and it also prevents contamination and erroneous results. This allows large samples of DNA to be processed in relatively less amount of time. So what does a qPCR machine look like? Here we can see a typical qPCR machine, which inside contains the thermal cylinder and light emitters and detectors to detect fluorescence data, and it can use this data to generate frequency of DNA gene expression or other targeted DNA sequences. Here are our credits for our sources.